Hello and welcome. Thank you for joining us for the third annual Black in Immuno Week, a week where we will celebrate, amplify, and support Black voices in immunology. Before we begin, we would like to thank our major sponsors who have made this week possible. You can find our full list of sponsors on our website. Don't forget to tweet about our session today using the hashtag Immuno Crossover, with each word capitalized for accessibility purposes. We will have the live captions and ASL interpreters during the session. Click on the CC at the bottom of the screen to access captions. You can submit questions through the comment set function on the platform, not the Q&A. As a reminder, we require all audience members to be polite and respectful while in the Black in Immuno space. If we see anything offensive, your comment will be deleted immediately and you will be removed from the platform. After the conclusion of the week, recorded videos will be posted on our YouTube channel and transcripts will be made available on our website, blackinimmuno.org. My name is Welt Okete, and I am a biochemist from Nigeria with keen interest in immunology. I am also host of the Immunology in Africa podcast. I will be the moderator for the basic immunology session today. We have five speakers here to tell us about their work. They have about 15 minutes for their presentations and will follow with a 5 minutes Q&A. Without further ado, let's jump on in. Our first speaker for this session is Dr. Cherie Butts. Dr. Cherie Butts joined Biogen in 2012 and she is currently medical director in research and development. She was clinical lead for PEG interferon beta 1A approved for multiple sclerosis in 2020 and in the US in 2021. She completed a health equity assignment focused on addressing gaps in clinical data with a goal of empowering patients to become more informed, to make more informed treatment decisions. Dr. Sri earned a BA and MS degrees from the Johns Hopkins University, a PhD from the University of Texas, MD Anderson Cancer Center, and she completed a postdoctoral fellowship at the National Institute of Health, studying neuroendocrine regulation of immune cells. Dr. Bott is passionate about connecting research across academia, government, and industry to advance the biomedical ecosystem. She serves on the board of several institutions and organizations, such as the Leadership Advisory Board of Beth Israel Deaconess Medical Center, Board of Trustees, Board of Directors of Keystone Symposia, Board of Trustees Chair of Salem State University. Welcome, Dr. Butts. You can proceed with your presentation.
Thank you. You can hear me? Yes. Okay, perfect. So I only have 15 minutes, and I thought that it would be better not to use slides, but to tell my story. Because at the end of this session, what I'm hoping is that you receive it as a call to action. You can read about my story, which is fairly unique because I've had the privilege of working in academia, as we all grow up, in government at both FDA and NIH. And also, as was noted, I'm currently in industry. And so the polls that you all received before, which were asking about your experience, those of you who have either worked the majority of your career in academia or in nonprofits, or if you've had the opportunity to learn about working in government and in industry. And then the second question is really about developing new therapies. And one thing that I have appreciated most about my time in both government and in industry is this new way of thinking, because the key questions that you're asking when you're in government and in industry are very different from the key questions we tend to ask in academia. The focus in academia is more on the fundamental biology and understanding of how the processes are, whereas for the government, it really is a public health organization. And so the experiments that you're designing need to be asking those questions about how relevant is this to the population and in particular, the US population. And so the third question that I asked about health equity and trying to understand that for those of us who grow up in or who operate in government and in industry, health equity is something that we have to do every day because we can't just think about our research in the context of people who live on one street versus the people who live in all other streets. And so we're constantly thinking about the generalizability of the work that we do. And as it goes in industry, when you're developing a new therapy, when you're developing a new diagnostic, when you're developing a new screening test, you can't simply test it on one group of individuals. You need to make sure that it's relevant for everyone who has that disease. And so for the work that I've done as a member of as a scientist working in government and as a scientist working in industry, we really start off with, depending on what the disease is, what's the epidemiology? Who has this condition? And something that has been very important to me because I recently did a health equity assignment is understanding the role of geography. I grew up in Texas. And so in Texas, I was outside all the time. There is plenty of sun. It's hot. I eat very much like a Southerner, which is, a lot of gumbos and barbecue and the like. But when I moved to Maryland, when I did my postdoc at the National Institutes of Health, and when I transitioned to the FDA, it was colder. I wasn't going outside as much, although Maryland is a very beautiful state. And so there was some level of getting out, getting on the water. And then I went to industry. So I went to Biogen, and that's in Boston. And I'm sure you all know Boston is cold. So I don't go outside as much as I used to when I was in Texas. I don't eat the same foods. They don't have barbecue and gumbo as I enjoyed when I was in Texas. And so thinking about the impact of my lifestyle on how I operate, I'm the same person as the one who lived in Texas, as the one who lived in Maryland, as the one who now lives in Massachusetts. But because I operate differently, because the foods I eat are very different, I need to be thinking about the impact of if I had a condition, if my lifestyle has changed, it could impact the outcomes of the disorder that I have. And so that is something that I've really enjoyed about the work that I do. And that is something that I've really been trying to communicate to others is we need to be thinking about the relevance of our research. And there's a tendency to say, oh, is this something that happens in Blacks, something that happens in Latinos, something that happens in Asian versus something that happens in Caucasians. But we know that Blacks are not a monolith. There's no one version of us. And so if you try to generalize something to say that this is relevant for Blacks without understanding all of the different ways that we are Black, then you've also done a disservice. And so I want you to be thinking about the research that you have beyond race, beyond ethnicity, because we are so diverse even within that group that we have to think about what are the other elements, the other factors that could be driving the health outcomes. And that's why I have a tendency to pick on geography because it's a little bit more neutral. As I mentioned, there's also the foods, there's also the lifestyle. Do we go outside more? Do we exercise more? Do we have access to certain resources and do we utilize them? And all of these things do play an important role in the research that we conduct and the outcomes from that research. And so one of the things that I've been really driven lately to focus on is this idea of time to diagnosis. When I was doing my health equity assignment, I purposely wanted to do research with Black and Brown for certain diseases like lupus, Alzheimer's, many of the disorders that are important for my company. And one thing I tried to convey is there's a tendency to interchange the word underrepresented and the term underserved. And I said, you cannot assume that every Black and Brown person is underserved. And so, in fact, when we conducted that research, I wanted to over index on underrepresented, so Black and Brown people who may be living with a certain condition who are not underserved. So they have commercial insurance, they have access to, they have resources, they have access. That is not their issue. And so the questions that we were asking is, you know, do you have barriers? Like why are we not seeing more people participating in clinical trials that come from these communities if we remove what tends to be highlighted as disparities, which is lack of insurance, lack of transportation, and things like that. So we over indexed on those individuals who, in fact, don't have those issues. And we said, okay, well, why aren't you participating in the clinical trial process? And I'm sure you will not be surprised that for the most part, the answer was nobody asked me. And what we did in our research is we did not just ask the patients and their families, because we recognize that family is just as important in the clinical trial participation process as the individual themselves, especially for those disorders where the population is older. But we asked these same questions to providers, to clinical trial coordinators. We even asked it to the payers or the insurance companies. And we said, okay, everybody gets the same question. And we want to understand, is the answer the same? And I'm sure you won't be surprised to hear that the answers were not the same. And what we found highlighted was this, I don't know if the right word is lack of communication or miscommunication, but I'll give you one very, very good example. We had an individual who had been diagnosed with Alzheimer's, and we asked that person, okay, well, what was your experience? And they said when the doctor gave the diagnosis, there was shock. Lupus, same question, same answer. So there was shock, and they had to process the information, and they were really they had a lot of questions, but to be quite honest, they needed time to let this diagnosis sink in. So then the flip side, we asked the provider, when you gave this diagnosis, how do you think the person responded? And the interpretation was apathy, that they didn't care, that they weren't really paying attention, they weren't really listening. And can you imagine what that room was like, where this person is going through this emotional state because they've just been told they have a condition, they have no idea what they're going to do with it. So shock is fair. But because they're not saying anything, it's interpreted as apathy. And so these are the kinds of missteps that we really do believe could be causing this dramatic delay in the time to diagnosis for those who are coming from Black and Brown communities. This idea that because the communication is broken down, where you might be getting, okay, we're going to do a screening, we're going to do additional tests, we're going to make sure we understand what this is. Because of that initial lack of communication and that breakdown, then sometimes when I say there's a delay, I'm not talking about a few weeks. We're talking about years. We're talking about 10 years, sometimes 20 years. And why is that important? So let's take something like Alzheimer's disease. This is something that there's a lot of investigation and development of new therapies. And when you look at those clinical trials and the therapies that are being developed, what stage of disease are they tending to target? Early. Everybody wants to hit it early. If you're able to target it early, then your quality of life is going to improve, because we know with early diagnosis and early treatment, it leads to better health outcomes. And so most of these new therapies that are being developed with the idea of either getting people who haven't started showing symptoms or who are early in the stages of disease. And therefore, you can imagine that if Black and Brown people are being diagnosed late, by the time they're getting diagnosed, guess what's happening? They're in the mid to late stages of disease, which means they're not qualifying for clinical trials. And so we have to be careful about saying things are because of the structural racism in the system, and we think about the practicality of that. If you're developing new therapies to target an early stage of disease, and yet the people who are you need to recruit for this trial, they're probably not going to be Black and brown, because Black and brown are taking too long to be diagnosed. And so this is one example of the work that I've been doing, where I've really been trying to understand, why do the trials look the way they do? Um, and so before I move into the next phase of what I wanted to talk about, I want to talk a little bit about the work that I've done. So I've had the chance. So I started off as a neuroimmunologist working at the bench, uh, doing immunology, thinking about how the reproductive and nervous system can contribute to the biological processes. And then I was a regulator, so I reviewed the new drug applications, and I was a CMC, which is chemistry, manufacturing, and controls, to make sure that the products that are being developed are then, every batch is going to be the same. And then when I came to Biogen, I started off in the research group, but then I realized that the one thing that I hadn't done, I knew concept, I knew approval, but I hadn't done the clinical development part. So I said, you know what, let me try clinical trials and see what that looks like. And what was really interesting when I transitioned into a clinical development role and designing the trials was, the conversation came up about the representativeness of trials, but we weren't really focused on it at the time. And so we would design the trial, have the protocols, meet with the investigators, and have them recruit, complete trials. And as was said in my introduction, I had the pleasure of taking one therapy all the way to the end and getting it approved in both the U.S. and the EU. And I will say that was a wonderful, wonderful moment for me, which I don't think I ever would have had that chance had I stayed in academia. So I would say that being in government, being in industry, it's just a different set um, of goals, and a different set of what can you do and what can you accomplish. And I've really appreciated that opportunity to say, that's my drug. I'm the person who took that to the finish line. But when this idea of health equity, and especially when the pandemic came up, the thought about the representativeness of our trials. And so my division chief said, well, why don't we see what our record has been, and so we know, how far are we from reaching the mark. And sure enough, when they went back and looked at the trials, they noticed that all of my studies were highly diverse. In fact, the one for PEG interferon that was approved was more than 50% Black and more than 30% Hispanic. And so there might be some Blacks who were Hispanic, but the point is, it was more than 50% of non-whites. And so they were trying to figure this out. And I said, well, I did not talk about representation in this trial, but what I do say is that I was in the room. And I can't say that if I wasn't in the room, that they weren't thinking about this. And so that is something that I really want you all to be thinking about, is sometimes when you enter the room, the conversation changes and you don't even have to say anything. And that's power. And that's power I want you to embrace. And so many times we'll be talking about the fact that, we want to go into the room and we don't want to change the conversation, but your presence alone can change the conversation. And that's a very important element. And so I have a few polls that I wanted to put up, and let's see if Susanna can help me out a little bit. But one of them is about decision making, because one of the things, as was read in my bio, that I've had the opportunity to do is serve on a lot of boards. And I recognize the power of decision making. And so I want you all to be thinking about your decision making experience, and choose wisely what committees you sign up for, because there are always going to be opportunities for you. I guarantee that. But you have to be ready for them. And sometimes what I've seen with my colleagues who look like me is they're over indexed. They're being they're serving on, they're serving the community, and they're serving on different committees, and they're doing a lot of work. They're doing their day jobs, and so they don't have a lot of bandwidth that's available. And one thing I tell people is you need to protect your bandwidth so that when the opportunity comes, you're available to take advantage of that. But I would say, please do strive for those that are decision making, because those are far more important than simply serving on a committee. If you can serve on a board of the same organization, that's where the decisions are made. That's where the policies are made. And so I would really encourage you to do that. And then think about the service that you do have. I mean, many of us work in our communities, and our communities can be scientific. They can be in our particular identity group. But think about what kind of service you're doing, and think about the scale with that particular organization. And so many times what I ask people is, are you only serving at your institution? Or are you serving on national and international boards or committees? Because that really is where you're going to be able to scale things. And what's even more important than that is not just serving on a committee or serving on a board, but becoming the chair. Because I will tell you, as someone who is currently and has served as the chair, you're the real decision maker. You are really driving the conversation, and people are looking to you as the leader in order to define the direction of that organization. And then the last thing that I wanted to say, and then we'll open it up for questions, is really about your sphere of influence. I will talk to many students, and they will say that they're thinking, okay, once I become a professor, then that's where, I can really have influence. But we all have influence right where we are. We all know our networks, and we know our networks among them. And so what we want to be thinking about is, how can I influence where I am now? Not, okay, maybe in the future I'll be able to do something when I become big time. What is it that I can achieve now? And the reason why I bring this up is because if you think about your own personal sphere of influence, and then you have a friend who does the same, and another, we don't have to boil the ocean. We can each do this together, and then we can take care of the little microcosm that we have access to. And if we all agree that we're going to make our space a better place, then it's going to be a domino effect. And therefore, I want you all to be thinking about that. What can I influence today? How can I influence it? And maybe can I get some ambassadors who can do it in their spaces as well, so that we're all doing this. And if you do this, if you speak up and stand up, and you do it for the right reason, and you make sure people understand that your voice is important, then that is going to truly change the biomedical system, the ecosystem. And look, by dividing it out and having ambassadors who are helping you in this process, you don't have to be exhausted, because you say, I'm going to take this part, you take the next, and we can help each other, but we also are taking little chunks. And so that to me is much more important. And I want you to be thinking about that now, but also in terms of your career aspirations. Because as you're thinking about what do you want to do next, you can leverage that same network and say, you know what, there's a couple of things I will tell you. I didn't expect to be doing what I'm doing now, but I had a network who saw more in me than I saw in myself. And they were willing to go out and say, hey, why don't you try this? And probably the most important element of all of this is that when I was given the opportunities to try, I could always go back. I could always go back to government. I could always go back to academia. And so none of this was really risky, because I knew that I could go back to where I was if I didn't like what I was doing. And so that is also what I want to encourage you to be thinking about, is your career aspirations can be so much bigger than what you're seeing in yourself, but your network can help you to think even bigger and even broader and have greater impact. And so with that, I think I'll close, and then I'll be happy to take any questions. I'm going to need some guidance, Wealth, on how to take the questions, but.
Okay, thank you very much, Dr. Cherie Butts, for your presentation. Um, I'm not sure there are questions yet, but if you have questions, you can quickly drop them in the comment section so we can take them. Okay, we'll
And
still wait for a few minutes before.
okay. And I was going to say, if anybody has questions, they can always shoot me an email.
Okay.
Or if we don't have any, I probably took all my time. So we can also transition to the next, and then maybe we can come back at the end.
Yeah, thank you
You
so
tell
much,
me.
Dr. Potts, for your
Okay.
great talk. Yeah, there are no questions now, so we'll just move ahead to the next session. Thank you
Okay.
so much
Thank you.
for sharing your experience with us. Yeah, so we'll be proceeding to the next session now. And our next guest is Dr. Catera Wilder. So Dr. Catera Wilder is an assistant professor in the Department of Bioengineering and Therapeutic Sciences at the University of California, San Francisco, and Chan Zuckerberg BioHub investigator. She received a PhD in biomedical engineering in 2016 under the direction of Dr. Manupat. She, as a UCLA Chancellor's postdoctoral fellow in the lab of Dr. Alexander Hoffman. She focused on understanding innate immune and inflammatory responses by studying ISDF3 transcription factor dynamic regulation using a systems biology approach. Our work investigating the interferon signaling and transcriptional regulation has uncovered stimulus specific responses determined by coordinated stimulus contingent positive feedback loops. She's interested in investigating the spatial temporal control of intra and intercellular interferon signaling networks and developing predictive quantitative tools of immune responses. You're welcome, Dr. Wilder. Yeah.
Hello, thank you for having me. I'm really excited to be here. And I'm looking forward to discussing a lot of the work that I've done as a postdoc. So I just started as a professor at UCSF a few months ago. So most of the work I'll be discussing is what I did as a postdoc, but looking forward to expanding this work as I continue on as a professor at UCSF. So I won't be able to get live feedback, but I wanted to start off by just showing this picture and trying to determine what people see and how do they interpret this particular image. And while some people may see a young woman who's looking backwards, where this is her chin and she's looking kind of this way, and this is just like a headpiece, others may see an old woman who's looking down. And while this is her nose and her mouth and also a headpiece. And so I think it's very interesting that people can look at one particular image and see two different things. The process of what's happening and how that's happening is very similar, regardless of what you see. And so there's some sort of data processing that's occurring to get either the old woman or the young woman in terms of an output. And so this data processing is just this conversion of raw data into something that was more meaningful. And so you have this input. Again, there's some type of processing, whether it's if you're thinking again of looking at a picture, you have some processing in your brain, but I'm interested in the immune system. And so a lot of times I think of cellular dynamics and the processing that's happening at the cellular level, and then what is the output that is occurring after that processing. And while the example that we used with the woman, the young and old women, there was no real consequences of picking either the young or old. You can imagine that during an immune response, there are some consequences that can occur if the cells receive an input and incorrectly distinguish what that input is. And so you can imagine that happening when there's a question of whether an antigen is viral or a self-antigen. And if that is incorrectly determined, you can have activation of autoimmunity. Same thing where it's important to distinguish a particular input when there's an infection, determining what type of pathogen is present, whether it's viral or bacterial. Again, there could be consequences if you incorrectly determine which one it is, viral or bacterial. And then the same thing, once your system or your immune system knows what type of pathogen, it still has to determine what type of immune defense mechanism should be employed. And again, there could be consequences depending on if that's incorrectly determined. And so how is this happening at a cellular level? And so most of you probably at some point have studied cellular signaling, where most cells have some sorts of receptors for various ligands. And so the information is input at this level where you have this initial receptor binding. That information is then propagated using a lot of signaling pathways and effector proteins. And so that's the stage of information encoding, where whatever was sensed at the receptor level is encoded through these networks. And then typically these transcription factors then translocate into the nucleus, where the information is decoded and it's determined some important biological function needs to occur. And so there's transcriptional regulation that induces transcripts, and then downstream translation and cellular responses. And so one question that I have is typically, how does this happen, and how do cells particularly effectively distinguish multiple extracellular signals? And so one way that this could occur, and a hypothesis I had for many of my studies, was that there was some dynamic temporal code, where a lot of times the cells in our system only have a particular set of receptors that can bind to multiple ligands. And so in that case, if you do have different ligand receptors that are activated, if they activate the same pathway, it could be done so at some type of temporal differences in temporal dynamic, which can help the cell to distinguish which ligand was bound. And so just a little bit more of what I mean when I say a temporal code. If these are just plots where the x-axis is time and the y-axis is some type of transcriptional factor activation, you can imagine that depending on the speed of the transcription factor activation, that could be one way that the cell can distinguish differences in ligand binding or ligand receptor activation. You can also imagine that the total activity of a transcription factor response can be one way that a cell can distinguish the type of ligand that is activating a particular pathway. And these are, again, just other examples of what could be happening that allows a cell to effectively decode particular sets of information. And so this is not a new thought. This has occurred, or this hypothesis has been seen and determined in a number of signaling pathways. And my previous lab that I was a postdoc in, the lab of Dr. Alexander Hoffman at UCLA, he studied this extensively with NF-kappa B signaling. And some of you may know that NF-kappa B is a very important transcription factor for pathogen sensing. It's activated downstream of a lot of the TLR pathways. It's also important for inflammation. It can also be activated by TNF. And so he has done a lot of work. Him and his lab had done a lot of work by the time I had started to understand temporal codes that are specific for different ligands that activate the NF-kappa B signaling pathway, but not much work had been done to understand how this could be happening for other pathways, and one in particular, this interferon signaling pathway. And so this question was definitely something I was interested in, because as you can see in this cartoon here, there's multiple interferon cytokines where you have different families. You have this type 1 interferon family where within it has about 8 different interferons, as well as the type 3 interferons, which even within that family have about 4. And so you can imagine that there could be importance in trying to determine which ligand, which interferon is actually activating this signaling pathway, especially as you can see here, these various cytokines can, they primarily activate this transcription factor ISGF3. And so can you get different outcomes or different cellular responses, even though you have all of these interferons that activate the same pathway? And if you do get different responses, how is this done? How is the information decoded so that the cell knows which interferon is present? And so just a little bit more background on interferons themselves. They're definitely important for immune responses. This is a Kaplan-Meier curve where mice were infected with influenza A, and they looked at their survival over time. So the x-axis is days post-infection, and the y-axis is survival. And they did this in wild-type mice, which are shown in black. They also did it with mice who were deficient for the receptors for the type 3 interferon pathway or the type 1. So type 3 is in blue and type 1 is in red. And you can see that when you don't have activation of this pathway because you no longer have the receptors, this greatly diminishes the survival of the mice that were infected with influenza A. So these interferons are definitely important for protection during immune responses. But it does seem as though there could be differences in how they are regulated. And so this was seen especially in this graph where these were COVID-19 patients, and prior to their entry into the ICU, they had serum that was taken, and they decided to measure their interferon level. So, and they took a ratio of a type 3 interferon, which was interferon lambda, and the ratio between lambda and alpha. And they saw that the amount of interferon lambda versus alpha ratio could be a predictor of hospitalization time in the ICU. And so as the higher ratios, you can see there's this negative correlation where higher ratios of interferon lambda to alpha was predictive of shorter hospitalization times, while lower ratios were predictive of longer times, indicating that the type 3 and type 1 interferons could be differentially regulated in disease. And so you could imagine that I had this question, which was, how are these interferons differentially regulated, or are there specific responses that can occur from these different interferons? And so this could occur by just cell type or tissue specific expression of the receptors, where you might have a subset of cells that express type 1 interferons and another subset that express the receptor for type 3. But I was really interested in a cellular system where you have expression of both of the receptors, the type 1 and type 3 interferons, and is there differences in responses for cells that express both? And then if there are, again, how are they differentially regulated? How is the information encoded? And so I decided to study epithelial cells. These epithelial cells are known to have receptors for both type 1 and type 3 interferons. And they're also some of the primary targets of infection, since they usually are at they're barrier cells. And also just understanding epithelial cells as regulators of immunity, I think, is really important, as these as immune responses are not unidirectional. You don't just have infection and then adaptive responses that occur later after innate responses, but you have this interplay throughout the time course of an infection, where you could have cells that are in your innate immunity responding to what's happening in the epithelial layer. And again, this crosstalk between just all cells within the immune response could be happening. So understanding how the epithelial cells in particular regulate that, I think, is really important. And so one of the first things that I did as a postdoc was I decided to incubate MLE lung epithelial cells with either interferon beta, which is a type 1 interferon, or interferon lambda 3, which is a type 3 interferon. And one of the kind of more striking things that you can see is this top cluster, where there's a cluster of genes that seem to be specific to interferon beta, but not interferon lambda 3, which was one of the questions we had, which was, are there just differences in the types of genes or different cellular responses that could be activated by these different interferons that activate the same pathway? And at least from this RNA-seq experiment, so I didn't clarify, this is a heat map of RNA-seq where each column is a time and each row is a gene. So you see that there are specific genes that can be preferentially activated by the type 1 interferons. And so we did some gene ontology analysis, and we did indicate or show that a lot of the genes that are in this top cluster happen to be genes that are involved in pro-inflammatory responses. And so I decided to measure the transcriptional activity when these cells were stimulated with either interferon beta or lambda. And so this is a mobility shift assay where we measured the ISGF3 transcription factor. And this is just one example, and this is quantification of multiple experiments on the right. And one of the things that is very obvious is that during the middle of this time course, you see this potentiated phase of ISGF3 activity where you get this hyperactivation during starting around 100 minutes that lasts till about 6 hours. And so that's not seen when the cells are stimulated with interferon lambda 3. And I'll just point out that at the beginning, it seems as though the cellular pathway is activated at similar magnitude. So within the first hour, they have similar activation, but something's happening in this middle of the time course that's causing this potentiated phase when the cells are stimulated with interferon beta. And so as a systems biologist, I decided to try to see if there were mathematical tools that we could use to try to answer or try to determine how this could be happening. And so I won't go in detail about the actual model itself, but these were simulations that were produced from the model. Again, it was a model of ordinary differential equations of the signaling pathway. And one of the things that it predicted was that dose was important. Dose and duration were important for causing this potentiated phase of ISGF3 that we had. And so in this particular graph, each color is just the ISGF3 transcriptional response, but at different doses where the dose is decreased as you kind of go down. And you can see that as you decrease the dose, this potentiated phase is no longer as pronounced. And that's also shown in this bar graph where we took the same simulation, but just plotted the response at 1 hour and 4 hour. And you can see at these higher doses, the potentiated phase seems to be more affected by just small changes in the dose, while the 1 hour time point seems to be similar when you look at these, at least these upper doses that are used. And not only is this potentiated phase seems to be sensitive to the dose, but also duration. So in this particular simulation, we had a short duration of interferon beta stimulation of only 15 minutes. And you see in the purple line that also prevented this potentiated phase that we had seen. And so we decided to experimentally test and see if our model was able to accurately predict some of these cellular responses. And indeed it was, where again, when we tested experimentally, the dose, as you can see in these two graphs, were definitely important in terms of preventing this potentiated phase that we saw, and also the duration. Again, when you only had incubation of the cells with 15 minutes of interferon beta, you prevented that potentiated phase activation. And so this is really exciting, and I'm happy we had quantitative tools, but we also wanted to see how does that play out in terms of how this information is decoded at the transcriptional level? Are there just any functional consequences? And so we then repeated the RNA-seq experiment with these new conditions of low, which is in light blue, as well as pulse, which is in purple. And you now see that the transcriptional response is very similar to what we saw with interferon lambda in red. And so this is seen in this PCA plot of the RNA-seq data, where the 4 hour time points of lambda, low pulse, and beta pulse all cluster kind of in this middle. But the 4 hour time point of interferon beta, the higher dose and duration, kind of clusters out here. And so there does seem to be this switch that turns off when you no longer have longer doses or higher doses or longer durations. And that's also seen right here in this heat map, where again, when you have this lower pulse, you no longer get activation of this cluster 1 genes, which again included some important pro-inflammatory regulators. And so we wanted to determine how this was happening. Up until now, we showed that the dynamics are important. And so again, you can have the same signaling pathway activated, but it seems as though the dynamics of how that pathway is activated is very important, but we want to understand mechanistically how is that happening. And so we decided to block some of the positive feedback loops based on this model simulation. So again, we use that quantitative model that I showed previously, and we just showed that, at least with the model, that blocking the positive feedback of STAT2 or IRF9, which were just important regulators of the pathway, could prevent this potentiated phase. And so we decided to use CRISPR-Cas9, where we block that positive feedback loop. And so this is just validation of the model itself, but it's also showing that, again, this is not a knockout of STAT2 or IRF9 itself, because at unstimulated time, you see that the amount of STAT2 transcript, which is seen in this plot, as well as IRF9 transcript, is similar in the mutants as well as the lenti control. But then after 1 hour of stimulation, that's where you see this downregulation of either STAT2, which again is seen on the plot on the right, or IRF9. So again, this is not a knockout of the protein or the transcript itself. It is just a blocking of the actual positive feedback loop. And so based on the model simulation, we decided to test it experimentally to see if we block this positive feedback loop, does it impact the signaling dynamics that could be used to activate these pro-inflammatory genes. And indeed, yes, it did. So this is experimental data of the actual transcription factor ISGF3. And we have these 2 mutants that are in gold or purple. And you see that when you have the feedback mutants, you no longer have this potentiated phase. But then also interestingly, when you just block that potentiated phase, we also saw a decrease in the amount of expression of particular genes that were seen to be important. So IL-6, especially when we have in this IRF9 mutant, it decreased the amount of gene expression. You saw the same thing with TNF-SF10 and some of these other genes that are important for pro-inflammatory responses. And so in conclusion, it appears as though there can be differences in these interferon cytokines, which are important for not just antiviral responses, but also for type 1 in particular, for these pro-inflammatory genes. And hopefully I've been able to show you that there's this dynamical code that is needed to distinguish the different interferons. And so while I showed this for this interferon pathway, again, this kind of a theme or thought could be taken or applied to other pathways as well, where it's really important for the cell to be able to provide effective information processing and information information encoding by regulating how the temporal code of these transcription factors are regulated. And not only is this, again, important just at the cellular level, but how this plays out in terms of function is important when we're thinking about just different inflammatory processes that are activated in the immune response, as well as any autoimmune diseases. And so I just wanted to thank a lot of this work, like I said earlier, was done in the Hoffman lab. So I wanted to thank Alex Hoffman, as well as people who helped with different aspects of the project, as well as a co-advisor, Erin Meyer, who was also at UCLA, and just the funding sources that helped fund the work. And then at this time, I can take any questions.
Thank you very much for your session, Dr. Wilder. So we have a question from Yasin. He's asking, do you account for receptor availability and internalization in your ODE model?
So the ODE model does not have receptor internalization, no, but that's definitely a component that could be added. And I think it brings up a good point that there are definitely just different ways that the pathway can be regulated. And I do want to look into how the receptors are trafficked in the future, but this particular model did not include that aspect.
Okay, thank you very much for your response. That's all for now. So we'll be proceeding to the next talk. Thank you very much, Dr. Wilder, for your Great talk. Our next speaker for this session is Dr. Dequina Nicholas. So Dr. Dequina Nicholas is an assistant professor at the University of California, Irvine, in the Department of Molecular Biology and Biochemistry. The Nicholas Lab studies the intersection of the nutrient environment, the immune system, and metabolic disease using a combination of molecular and cellular biology, transgenic mouse models, cytokine profiling, and flow cytometry. Dr. Nicholas received a PhD in biochemistry from Loma Linda University and pursued postdoctoral training in the lab of Dr. Barbara at Boston University. She studied the metabolism of immune cells from patients with type 2 diabetes. Dr. Nicholas also trained in the laboratories of Drs. Mark Lawson and Pamela Mellon, where she established the importance of glucose metabolism in reproduction and discovered a population of immune cells in pituitary gland that regulates the reproductive axis. Thank you very much, Dr. Dequina Nicholas, for joining us. You can proceed with your presentation now.
Okay, thank you so much for that great introduction. Everyone can hear me great, right? Microphone's working? Okay, so we're going to shift gears a little bit. I am going to give you some like spattering of data that I think is really, really cool from a great project that we're having. But one thing I really do want to talk about is a little bit of my vision, how us basically giving back to our community, driving independence, also using the power that we have in our spheres of influences, kind of jumping off of what Dr. Cherie Butts talked about, has accelerated my career. It can also hopefully accelerate yours. So, of course, all of you are very, very familiar with academic career stages. Go from high school, undergrad, maybe a master's or postbac, graduate school, you do your postdoc, then you're faculty, but you're pre-tenure, job's not secure, then tenure and beyond. So what's crazy about this whole academic pipeline is that earlier on you are in your career stage. Of course, the more new ideas you have, the more energy you have, but this is when you have the least amount of independence and the least amount of power. And then as you progress through these stages of your career, you gain more and more independence and more power. So really what I want to stress is the ability to use the sphere of influence that you have at each of these distinct stages in your careers to actually exercise your independence, use your power, not only to help accelerate your career, but to help give back to the communities from which we've come. So with that said, I'm going to start off by just talking about my laboratory. So my lab, brand spanking new, maybe not brand new, but it's been established for about a year, opened up last year in July 1st. So what is it that the Nicholas Lab does? We study two diseases that for me are very personal. They're important because they run in my family. They're also highly prevalent in Black communities, also in Hispanic and underserved communities. So we look at polycystic ovary syndrome, which is a multi-parametric syndrome that basically comes along with reproductive abnormalities. It causes the highest incidence of infertility for women that are in reproductive age. And we also study type 2 diabetes, which if you are Black or African American, then obviously you understand that there are very, very high rates of type 2 diabetes. Sometimes I call it T2D for short, in our communities. And what really ties these two diseases together, these two endocrine diseases, is that we know there's chronic inflammation underlying both diseases. In polycystic ovary syndrome, mechanisms are less known, and we're not going to focus on that for the small project that I want to share with you today. But we do know that it is a big deal in type 2 diabetes. And for almost 2 decades now, we have literally been studying chronic inflammation, characterizing it in type 2 diabetes, yet we have not made any progress towards treatment. So right now for diabetes, the status quo is basically we're going to manage symptoms, right? We tell the patients that everything about their culture sucks, is wrong and unhealthy, go eat some quinoa and walk, and then wonder why diet and exercise interventions don't help. Because we are not actually directing our interventions in a way that will help the communities that are actually affected. So like I said, chronic inflammation is actually an important part of the disease development, but there are no therapeutics or interventions that are anti-inflammatories to help ameliorate type 2 diabetes. So why is it that we haven't been able to leverage the chronic inflammation that we know or the problems that we know? Part of the reason is that there's 3 outstanding questions that just have not been answered in the field, right? We know the inflammation is there, but 1, we do not know the cause or the source of the inflammation. And number 2, we are not very clear on how chronic inflammation can contribute to disease. So then that last question, number 3, of how do we leverage what we know about inflammation to treat disease, it doesn't translate because we're missing some of the important indicators of disease. And another big hurdle is that in the field, we love immunologists in general. We love our mouse models, right? Of course, we get whole animal physiology, and the immune system is very similar to human. But at the end of the day, if you take a mouse, stick it on a high-fat diet, a mouse is not a human, and a human is not a mouse. And therefore, we can lose many insights that are very important by basically solely studying mouse biology instead of making sure that we're answering questions about human disease and human immunology. So what we've learned thus far is that inflammation in humans with type 2 diabetes looks very, very different from that in a mouse. For the most part, it is driven by a T cell-centric inflammation that is Th17. And then in the mouse, we see that it's mostly Th1 driven. So for example, in humans, of course, you have this response where if you take human PBMCs, take them outside of the body, stimulate them in vitro, they will secrete a whole slew of cytokines, one of these being IL-17. You also see both forms, IL-17A and F, you see IL-21, and then you have a cytokine, what we call profile, that is very, very indicative, and you can actually predict diabetes status based off this immune profile that's secreted from the T cells of people with type 2 diabetes. We do not see that in the mouse. What we've also found is that if you take B cells out of this whole mix of immune cells and you remove the interaction of B cells with T cells, then we lose that response that looks like it's Th17 driven. So this tells us that B cells are also playing a very, very important role in human type 2 diabetes, which we have seen in some mouse models. So I sat here in grad school, and it just kind of like, it just didn't sit well with me. I'm like, what is causing this type of inflammation? And so the field has searched and searched for antigenic drivers. What is turning on the immune system? How are we getting all this long-term chronic inflammation? And it occurred to me, one of the greatest risk factors for type 2 diabetes is obesity, high-fat diet, which is why we use the high-fat diet mouse model, right? And I was like, well, how come lipids aren't considered as maybe a potential antigenic driver of type 2 diabetes? It's elevated in diabetes, whether you're lean or obese, but as long as you have type 2 diabetes, you have elevated lipid levels. And then so I started digging into the literature, right? And I found that there's this whole class of molecules very, very similar to MHC1, MHC2 that can actually present lipid antigens. And these class of molecules are called CD1A. And I found that these type 1 CD1, CD1A, B, and C are not expressed in mice. So of course they get overlooked because the models that we focus on for studying diabetes don't have the molecules necessary or required for presenting lipids like we see in humans. So that made me think, you know what? I think that lipids could actually be important antigens for type 2 diabetes. And then so my overarching question is the immune response in type 2 diabetes driven by lipid antigens? And this question kept me up at night for years and years and years and still plaguing me, right? So the first time I thought about this was in grad school. I posed this question to one of my mentors, and I posted at a poster session, and I got laughed at by people who were senior in the field, who were immunologists, and it's like, okay, that's not how the immune system works. We were taught that classically small dietary lipids are not immunogenic. They don't drive an immune response. And that hurt, that stung to have my ideas just kind of like pushed to the side. But as I mentioned, no matter what career stage you're at, you should use what independence you have and the power that you have to either accelerate yourself forward and to help others come forward with you. So I applied for a really, really small internal grant at my institution, and I got the money. And I was like, you know what? We're going to use this money where I actually have control over experiments that I want to do. And I'm going to try out the hypothesis or test the hypothesis that I have. So me, along with some summer undergrads, we ended up building an approach that eventually got published and was the basis of my entire dissertation. So what was great about this was that I was able to use my background in organic chemistry and intermix that with the training I was getting in immunology. And we literally built an anti-lipid ELISA. So what we did was we took some saturated fats, particularly palmitic acid, which as we know is saturated, it's unhealthy. It's not good to have that in the diet or circulating free in our blood system. By using basically some basic organic chemistry, well, to me it was basic because that was my background, we took this palmitic acid, conjugated it to bovine serum albumin, and then used that protein anchor to attach it to the bottom of our ELISA plates. We then ran our ELISAs, and we were able to show that we can detect IgG class switch antibodies that recognize specifically anti-long chain saturated fatty acids. So this is the first time anyone was able to show that our bodies are somehow creating antibodies that recognize these dietary lipids. That was a big deal. That's a big deal for me, and it's going to be a big deal for the field, so look out. So what was also really cool is that we found that this also came along with changes in type 2 diabetes status. So those people in the cohort who had uncontrolled type 2 diabetes, right? Their blood sugar was through the roof. They had high A1C. They weren't exercising. They hadn't seen a doctor in like ever, basically. Once we measured the lipid or the anti-lipid antibodies in their blood, we saw it was super high, super elevated. After a 3-month intervention that was culturally sensitive, basically took into account the types of food that they eat, like what they have access to, their resources, the spaces that they have for exercise. We taught them how to take their blood glucose. After this intervention, we were able to show that the levels of these antibodies came down, meaning that it's changing with diabetes status, so it must play a role in onset of the disease. So moving forward, I thought like, okay, this paper, I published it. I want to continue pursuing this. In my postdoc, I once again kind of brought up the idea that, hey, I would like to pursue this. I would like to see if in type 2 diabetes, there is an immune response generated against lipids. And once again, even after publishing this, I was actually laughed at in my face. I got told, why do you like, it would be stupid for our bodies to basically generate an immune response against dietary lipids. Like, what would that do for us? What would be the purpose? What would be the point? And then so like I took my ideas, put them in my pocket, but I kept on moving. I kept on working on them, right? I kept on taking these ideas and formulating them together. So after my postdoc, when I got my job here at UC Irvine, I decided, you know what, I'm going to put in my ideas and hopefully it has to stick somewhere, right? I have evidence that these lipids could be driving determinants of type 2 diabetes. And if so, this would change how we think about diabetes as a whole. This would actually give us support for type 2 diabetes being an autoimmune response to dietary lipids. And in fact, this would actually be a unifying hypothesis for all the ways that we believe that type 2 diabetes onset is occurring. This would unify the ideas of chronic inflammation and insulin resistance. This would unify the ideas of leaky gut and endotoxemia turning on the immune system because they basically mimic lipids as antigens. This would also unify the idea that genetics falls behind type 2 diabetes. So of course, we could easily have differences in our genetics of how we respond to different lipids in response to diet. So what I decided to do, I took this idea, wrapped it up, I put it in as a grant, and NIH looked at it, and someone saw my perspective and was like, you know what, this is worth funding. So now, with a whole bunch of money in hand, we decided to move forward and press on with this project. So this idea that I thought of 10 years ago as a grad student, I never let go of it. I decided to keep on pushing, being resilient, and building a path forward. I was finally able to get funding for it when I was independent investigator. So in my laboratory, I have an amazing undergrad named Josue. He was here for a Bridges program as he was transitioning from community college to transfer over to a 4-year degree granting institution. So over the summer, I was like, okay, we have evidence that we can basically detect antibodies against saturated fatty acid, palmitic acid, but we have no idea if we can do any other lipids. We had to create this whole entire ELISA, but we want something that's more simplified that can be more high throughput. So we came up with the idea together to do what we call a lipid dot blot, right? So what he did, in a sense, was take Nutshell's membrane, very similar to a Western blot. He blotted them with same concentrations of different saturated fats. And we started with saturated fats because we knew we had the most likely chance of finding something that reacted within the plasma of these people with and without type 2 diabetes. So we did from carbon C6 to very, very long chain C24. What he was able to show is that indeed he could get signals of IgG antibodies that recognize lipids, just plain lipids in the serum. So when we did a whole entire panel of healthy versus those with and without type 2 diabetes, some very, very cool new biology has actually emerged. We see that in general, despite disease, if you have short chain lipids, you do not get an antigen specific response against these short chain saturated lipids. But when you look at long chain saturated fats, you see that we are building an antigen specific immune response, which has not ever been shown. So what I thought was really, really cool is that even just with the cool biology, yeah, that's all great, but there is actually changes between healthy and type 2 diabetes. We see that in type 2 diabetes, the very long chain saturated fats, the ones that are considered the most unhealthy that could be generated from a high fat diet, those are increased or elevated in those with type 2 diabetes as opposed to healthy. So we're also interested at taking the small cohort that we had as a pilot and looking at our IgM antibodies. Those aren't yet class switched, which would be basically our natural antibodies in circulation. And then what was really cool is that we saw kind of this flip phenotype, where for IgM, we actually do see a lot of antibodies against our short chain saturated fats, but not the antigen specific antibodies as we saw with IgG. And then for another comparison, you can see for IgM that these very long chain, you have lots of IgM for the healthy. but you don't have loss of IgM for the diabetic. And of course, this flip-flops in IgG. So I thought that was super cool and actually gives us a really strong basis for determining what is the natural antibody response to lipids in humans. So to do this, of course, we're going to have to expand, right? We need to screen those 10 free fatty acids aren't the only lipids that we derive from our diets. In order to expand, we want to create what we call a lipid microarray. So we decided once again to do some more what we call now crossover immunology and engage Dr. Ronki Olabisi. So she runs an engineering lab at UCI. And basically what we're doing is adapting a piezoelectric printer so that we can do very, very small drops of lipids and create a microarray. And then what's going to happen is that we're going to have to use some good engineering approaches. And we are basically 3D printing PVDF onto glass slides, and then using these slides to build this lipid microarray so that we can probe for hundreds of thousands of different lipids and see what is the antibody reactivity in both health and disease and with age. The other thing that we're doing in our laboratory is now that we know that these antibodies are being secreted, of course, the B cells have to be there. And we want to know what is the function of these immune cells that are recognizing lipids. One way to do this is by us building fluorescent probes on dextramers that by using click chemistry, one of my master students, Pawnees, is essentially taking saturated fats, biotinylating them, and then using that biotinylating lipid to adhere to streptavidin, which would be on this fluorescent dextramer, whereby we can sort any type of B cell that is directly recognizing these lipids. So in using all of these really cool approaches that are basically utilizing several different fields of science to answer our questions, we do have an end game in mind. Our end game, of course, at the end of the day, is going to be to cure type 2 diabetes and just stop managing it and managing the systems. So the biggest thing we got to do first is be able to figure out what are the lipid antigen determinants of type 2 diabetes. And we're going to need to do this by using this array that we've just discussed and that I showed you. So this is a mix of immunology, chemistry, physics, and engineering. Once we have these tools in place, we of course want to block type 2 diabetic specific inflammation upstream in physiological models, and by using humanized mouse models and also models that have immune cells in people with without type 2 diabetes. So all in all, my take home from all of this is that when people tell you no, push the boundaries, find a way. You have an idea, you have a unique perspective. It's important to use it and to exercise that power that you have. If the technology for the question that you want to answer doesn't exist, build it. Find collaborators. There's so much to our network here in Black and Immuno. There are plenty of people who can help you get to the goal that you want to get to as long as we do this together. And thirdly, like I said, just trust your perspective. Trust that your voice matters, that you have a unique approach to sometimes age-old questions that people are just too busy doing the same approaches in order to answer, which is why we're not getting anywhere. So with those three things, just keep those in mind. Remember, don't take no for an answer. Build the tools that you need and trust your perspective. So I just want to say thank you so much to my laboratory, everyone that helped me get set up, all of my funding, all of my collaborators. It's been such a blast pursuing my own independent ideas, and I just say look out for me in the future, and I'll take anything, questions that you have to ask at this point.
Thank you very much, Dr. Nicholas, for your presentation and inspiring story. Thank you so much for sharing it with us. So I would ask a question about your research, and my question is, you were talking about the challenge of the presentation of lipids as antigens in diabetic patients. So I just wanted to find out if non-diabetic patients is it possible for non-diabetic patients to have such lipids, and can that be a challenge for non-diabetic patients who may have, would that be a challenge for non-diabetic patients who could have similar lipids in their bloodstream?
Yes, of course. So there's going to be a big complication trying to compare like lipids from people who aren't diabetic to are type 2 diabetic. And what we really think is happening is that there's a threshold. So say if you just think of this as your general immunology and you get exposed to some type of antigen, right? The whole population got exposed to COVID, so all of us have COVID antibodies, but somebody who's actively undergoing an infection will have high levels of these antibodies, right? And there's higher levels of antigen circulating. So we think it's the same thing with the lipids, that all these lipids are normal. It's healthy. It's healthy to have these antibodies here. But if you have out of control levels and they're too high because you had a high fat diet, then of course we could push the immune response to becoming pathogenic, right? And then we have high levels of antibodies people with type 2 diabetes. And that's kind of how we would differentiate the two, is by levels, necessarily as whether one exists and another one doesn't exist.
Okay, thank you very much. You have another question from Dr. Daniel. Is asking one is asking is a ratio of IgG and IgM to long chain fatty acid in circulation a potential biomarker for the development of type 2 diabetes?
I think it could be. So one of the things we're really interested in is doing is that once we get this lipid array up and running, we actually have a cohort of people who we have followed over time and have all their serum collected. We have those who over time have developed diabetes and those who haven't. So what we want to do is track how does the antibodies change in the serum over time, and are there any indicators that we can find that are indicative of that switch from being either healthy to pre-diabetic to type 2 diabetic, or as opposed to progressing and not getting type 2 diabetes. So we think that could be a really, really good indicator, especially since we're seeing this nice, strong switch.
Okay, thank you very much. Thank you very much for your presentation. Okay, we have another question from Elaine. She's asking, have you or can you look at lipid reactive T cells in humans with diabetes? If yes, does it match your antibody data?
Okay, so yes, we can, because we are also building the tools to look at T cells. So of course, what most people will do is use CD1A tetramers, and that's actually available through the NIH. They work really well with human cells, don't work as well with mouse cells. And what we're in the process of doing is trying to stimulate and look at the cytokine profiles from those with type 2 diabetes. We haven't yet gotten to the point where we're able to match up the antigens that we see between T cells and B cells. But in my mind, I would assume that they would match, but time will tell.
Okay, one more question from Dr. Yassin. Can you use your lipid screen to detect people at risk?
I think we could. Of course, we have some a collaboration with some bioinformatists, and they say they will help us build a model for prediction. And I think once we build a model for prediction, we will be able to take our lipid array, basically take like 1 to 2 microliters of plasma or serum, and be able to say like, hey, this person is at risk of developing type 2 diabetes. That's part of the dream and part of the longer term goal.
Okay, so I think this will be our last question. Do you think the anti-lipid antibodies are a cause of type 2 diabetes or a consequence of the underlying pathological process? This is from David.
Yeah. So me personally, I think it's a consequence of the underlying process. What I really believe is happening in type 2, and you can quote me on this, is that we're getting activation of our antigen presenting cells, right? We're taking these lipids, we are educating T cells, and these T cells are basically pushing the differentiation of naive T cells to Th17 cells, and it's the cytokines secreted by this pro-inflammatory cascade that is pushing and causing insulin resistance. What I really believe these antibodies are doing is trying to reduce the amount of lipids or lipid burden that is circulating. So I believe the body is seeing these high levels of saturated fats as like invaders or as non-self, even though it is like a self-lipid, and is trying to reduce them by essentially using the antibody response to take them out of circulation. And the reason why I think this is because I have done experiments whereby we can use these antibodies in vitro, and it actually neutralizes the ability for, say, palmitic acid to activate dendritic cells in presenting antigen.
Okay, thank you so much for your great talk, Dr. Nicholas. We are glad to have you join us. Thank you so much. So we proceeding to our next talk. Our next speaker is Dr. Karissa Culbreath. Dr. Culbreath is the section chief and medical director of infectious disease at Tricor Reference Laboratories. She received a PhD in microbiology and immunology at Vanderbilt University and completed a postdoctoral fellowship in medical and public health microbiology at the University of North Carolina at Chapel Hill. She specializes in infectious disease diagnostics. Her research interests are in machine learning in laboratory automation and increasing health equity through access to laboratory diagnostics. Thank you for joining us, Dr. Culbreath. You can proceed with your presentation.
Thank you so much. It's very glad I'm very glad to be here, and wow, a big challenge ahead of me following these amazing presentations that have come before me. And though my initial training was in immunology, I've switched over. I spent a lot of time with the host, and now I'm over with the pathogens. So thank you so much for allowing a microbiologist to join you all for Black and Immuno Week. But I think we're siblings or cousins or something, and so it's a pleasure to be here with you all. I wanted to share just a little bit about me and my journey, because I think, of everything that you'll gather from the talks and the presentations today, I hope that everybody will see that there's a path for them in so many fields, that science and the science that we love is so incredibly broad, and there's so many paths for you to be able to take. And part of being able to find your path is hearing about the journeys of others, so that you can understand what may be available to you in the future. So I received my undergraduate degree from Fisk University and in biology, and then went on to Vanderbilt, where I got a PhD in microbiology and immunology. Have always been interested in the host pathogen interface, and so my PhD was more on the immunology side. But then when it came time for my postdoc, I decided to move over to the pathogens and get some space over there. And so I was at the University of North Carolina, Chapel Hill, for a medical microbiology postdoc through the American Society for Microbiology. And so it's 2-year training in medical and public health microbiology. That enables and prepares you to be able to run a clinical diagnostics laboratory. And so completed that postdoctoral fellowship. It's a 2-year fellowship, and there's a board exam as well. And so I passed the board exam, and then I came to Tricor Laboratories in Albuquerque, New Mexico, where we are the largest diagnostic laboratory in the state of New Mexico, and we provide laboratory diagnostics across the state. And so I've always been very, very passionate about being able to serve communities, and service truly is at the heart of everything that I've done. And so throughout the course of my education and training, there was always this connection to serving the communities where I live in. And when I was at the University of North Carolina, I completed a 2-month rotation in Malawi, which is in Africa, and I was helping with diagnostic development for HIV, malaria, tuberculosis, and routine bacteriology diagnostics, and really thought that I was going to spend the majority of my career working on diagnostics in sub-Saharan Africa, and spending a lot of time with capacity development there. But I found myself at Tricor in Albuquerque, New Mexico, and was feeling a little bit unsure about how the path was going to get back to where I wanted to be. And yet I found that there was still so much opportunity for laboratory capacity development and serving communities right in my own backyard. And so I started to dive into the work, and so I'm going to share a little bit about how the work that I'm doing right now around health equity and health access, using the COVID-19 pandemic as an example for how we are reaching out into communities in the state of New Mexico to be able to provide access to diagnostic testing. So I think many of us are quite familiar with this concept of health disparities, and health disparities being this very broad terminology that we use to describe not only the health conditions, but these larger preventable differences in groups of people that really impact their overall health. When we think about health disparities, we recognize that where people live, play, work, and learn have a bigger bigger impact on their health than even at sometimes their genetics and the other factors, innate factors of these individuals, that these social experiences really impact the overall health. And so we have this space of these health disparities, and what we're really trying to get to are spaces of health equity and health justice. That health equity is that everyone has a fair and equal and just opportunity to be as healthy as possible. And that in the space of health justice, that we want to ensure that people have the chance to be free from the things that would jeopardize or prevent them from experiencing their full health, but not only just that they experience their full health, that they have access to the ability to fully participate in society. Because what we recognize is that when we are unhealthy in our bodies and when we experience inequitable access to health, it not only hinders our ability to be healthy, but it hinders our ability to participate in society. And it hinders one's ability to be a fully involved member of society, and then that can have generational impacts throughout communities. And so this space of identifying how can we work in eliminating health disparities so that we can achieve health equity and health justice is something that I've been very, very passionate about. And often the efforts that we see in traditional clinical and in traditional medical spaces are those at the top of this health pyramid, where we look at the individual's response and the individual's responsibility towards their own health. But what we recognize when we start to get into a better understanding of health equity is that the underlying and the underpinning responses and the underpinning structures towards health are these larger context decisions, right? So changing the individual's context to make a healthy decision, or looking at the social economic factors, the community factors, the societal factors that truly impact one's health, truly make a difference in improving the overall health of an individual, but then even beyond that, the health of a community. But I work in the clinical laboratory space. And one of the things that people challenge all of the time is that as a clinical laboratory scientist, we are not often patient facing. And so much of the work around health disparities and health equity and health justice has been in the space of the patient facing clinician. So the clinician that is having the consultation, the clinician that the patient sees. But in the clinical laboratory, we are kind of the hidden heroes of healthcare. That there is a saying that the clinical laboratory is responsible for 70% of the clinical decisions that are made by a provider. And yet the clinical lab is just now coming into a position to begin to engage in health disparities and health equity. And why do I believe that it is important that the laboratory steps up as an active member in discussing and engaging health equity and health disparities? Is that there was a 2013 position paper where I have it highlighted here, but that the clinical laboratory is the most cost-effective and least invasive source of objective health information. And I think that's really, really critical when we start to think about many of the inequities that happen in healthcare and the implicit bias that we know that we see and experience in healthcare, that clinical laboratory data can be a source of unbiased health information. But that health information has to be reviewed and assessed and understood by individuals that may harness some inherent bias. And so one of the pieces of work that we do is help to repackage the health information that's provided through a lens of health equity and through a lens of health justice, that when an unusual laboratory value is identified for a particular person that may reside in a particular group or class or societal structure, that we try to make it difficult for a provider to miss it. We try to make it difficult for a provider to not recognize that this value is significant for this patient. And so we are trying to go from just providing a laboratory result to truly providing actionable health information. And the actionable health information that we are able to provide as a clinical laboratory is important in disease prevention, diagnosis, and ensuring healthy patient outcomes, patient safety, and participating as a member in public health surveillance. So when we start to think about that within the context of the COVID-19 pandemic, we recognize that the laboratory truly played an important role in the pandemic, but we also saw some of the challenges that the laboratory faced in being able to respond effectively to the pandemic. So when we think about a single laboratory result, we often talk about laboratory as just, I went and I got my blood drawn and I have a result. My result is interpreted by my doctor. I may get a prescription. But what we know in our laboratory, specifically, but the laboratory society and environment in general, is that the results that we provide are more than just a single laboratory result. We're able to look at not just the individual, but we can look at the individual over their longitudinal experience. We can have data from birth all the way into older age. And so we constantly are going through these cycles of screening, diagnosing, monitoring, treating, and so on for a variety of health conditions and a variety of health experiences. And so this longitudinal data gives us opportunity to be able to assess where an individual is right now, but also predict where they may be in the future and over time. And then we have the individual who's not just a single individual, but an individual that's in context of community, an individual that is in the context of society. And with that laboratory data for the entire population, we can then engage in activities of surveillance and prevention at the community level. But we also have additional data beyond just the laboratory data for that individual. In many cases, we have regional data. The laboratory may have access to address data that then begins to enable us to look more at populations and communities and understand that while there may be an at-risk individual for a specific condition, as a single individual, we can also, going back to our understanding and our framing of health disparities, recognizing that where people live, work, play, and learn have a huge impact on their health, that an individual that may be at risk for a specific condition may also represent a community that may be at risk as well. And so with this data during the COVID-19 pandemic, we really began to think about how can we understand data about communities and data, bringing in the laboratory data for those communities to be able to impact the disease progression. So during the pandemic, we certainly recognized that testing and laboratory diagnostic testing was pivotal during the pandemic. But we also saw disparities in that. There was a study in 2020 that found that the median travel time to a testing site was 20 minutes. And maybe that sounds okay for most people. Most people can probably travel 20 minutes. However, what they found even beyond that was that percent minority and percent uninsured and rural communities had a higher and longer travel time to a testing site. And not only that, there were challenges in accessing testing sites through public transportation. So 20 minutes by driving can easily be an hour or more if public transportation is required. And so this decreased access to testing sites resulted in increased demand for testing in communities of color, in uninsured communities, and in rural communities. And we know that these were the exact same communities that were more affected and more impacted by COVID during the pandemic. And so while a 20-minute travel time, we also saw these lines during the pandemic. And so when we're thinking about now an individual that may have had to take public transportation and that 20 minutes was really an hour, and then they were having to wait in even longer lines, when we have communities that may have resource challenges, that may have access challenges, that lacked paid time off to be able to get tested, then this barrier is not just a 20-minute travel time barrier. This barrier is an all-day barrier, and an all-day barrier then begins to turn into lost wages, begins to turn into lack of access to all of the things that communities needed to be able to stay healthy. I think the other thing that's important to recognize is that poverty was deeply associated to increased infections, increased hospitalization, and increased deaths. And so this data isn't for you to be able to see it. It's very small, but what I want you to understand is New Mexico is the fifth largest state in landmass, but is incredibly rural in population and also has incredibly high poverty rates. And so what you see is that each of these graphs represents increased amounts of poverty and increased amounts of COVID infection, COVID hospitalization, and COVID death. We also saw that these were the same communities that lacked access to testing. And so what we knew during this pandemic was that laboratory diagnostics and testing was key to prevention, especially early on in the pandemic when there weren't as many treatment options and there weren't vaccines available. So we needed to get people tested, but the communities that needed the testing the most often did not access and did not have access to the testing. And so that resulted in increased cases, increased hospitalizations, and increased rates of death in these communities. And so what we are doing right now is we are using the insights from communities to be able to develop. We've developed a proprietary algorithm where we are able to look at basic census information of communities as well as COVID-19 testing rates within those communities. And we are able to go into communities that are at risk, at community centers, parks, places of worship within the neighborhood and the community where the people are, where we are observing COVID hotspots. We're able to do the same thing in communities that lack access to testing, where there may not be enough testing for us to know what's happening in those communities. We're able to go into those same type of places and offer rapid, free testing during times where people need them and will be there. Offering testing during the middle of the day when individuals may be at work is not optimal. So we're able to offer testing at times where people can access their testing at the places where they are already engaging and already working. So we're taking this model and we're moving it across the city of Albuquerque and then moving even beyond the city of Albuquerque into other cities so that we can provide access to testing. And so this is a slide that we see often when it comes into conversations around equity. And when I think about diagnostics, and that's the space that I operate in, oftentimes we're hearing about new diagnostic methods, the most, you know, uh, emerging methods, and often these emerging methods are expensive. Often these emerging methods require extremely advanced science and scientists to be able to perform the work. These tests for emerging pathogens, we saw during the pandemic that access to testing, and we needed testing. And when we only think about providing testing in the traditional spaces and through the traditional methods, we still will often leave the communities with the greatest need, we will still leave them behind. There will be a test that they won't be able to access. And while the test might be able to provide them the diagnosis that they would need, if they can't access it, they are still behind. And so I really have been focusing my efforts on really thinking about what does testing equity look like and what does diagnostic equity look like. And so for me, diagnostic equity is thinking about what are less expensive methods to be able to provide accurate diagnosis. What is outreach into communities and not just centralizing the testing into to the traditional spaces around hospitals and healthcare, where there may already be distrust and well-deserved distrust. How can we provide rapid methods to communities so that they come on site, they're there for 10 minutes, they get their result, and they can make the interventions and the changes that they need. So moving from new, amazing, wonderful diagnostic methods, which are necessary, but bringing them to the communities and to the regions and the neighborhoods that need them most is really the direction that I've been focusing my efforts on so that we can truly get to a space of diagnostic equity. And so thank you so much, everyone who is working on diagnostics and working on the next steps and the next phase of the methods that I'll be using in my clinical laboratory. Make sure you keep these things in mind so that we can make sure that we are reaching the communities with the greatest need. Thank you all so much for your time.
Thank you very much, Dr. Culbreath, for your presentation. Thank you very much for sharing your work at Trico Laboratories with us. So the question I have now is, perhaps you could want to share some of the ethical issues that come up in your efforts to reach out to these communities or people who do not have access to standard health care.
Yeah, I mean, I think it's an ethical, the ethical issue is that they don't have access. And so I think we tried to balance that by ensuring that they have access. All of the work that we do work through IRBs to ensure that we're doing everything appropriately. And then we collaborate with the spaces where individuals are already engaging. I think one of the challenges that we know is that specifically communities of color, but many communities, especially rural communities and lower income communities, have a distrust of healthcare and the distrust of healthcare system. And quite often, unfortunately, it's a well-placed mistrust that healthcare has often left them behind or not responded to their needs appropriately. And so our work is going to the places that have already built trust and relying on the trust of those organizations. And it's something that I hold very, very dear and very sincerely, that when I engage a community center or a place of worship or a lodge or other community partner, it is my responsibility to retain the trust that they have built. And so we go into those communities very carefully to ensure that we are able to maintain that trust and provide that access.
Very much. Thank you very much for your response.
Mhm. Thank you.
So, in the absence of further questions, we'd just like to say thank you for your great talk and thank you for honoring our invitation. Yeah.
Thank you.
Our next speaker for this session is Dr. Daniel Abebayehu. Dr. Daniel is currently a postdoctoral fellow at the University of Virginia. After receiving his BS in biomedical engineering from the University of Virginia also. So he went on to receive a PhD in biomedical engineering from Virginia Commonwealth University, VCU, where his dissertation was focused on developing immunomodulatory biomaterials. So Dr. Abebayo is currently a postdoctoral fellow at the University of Virginia in the Department of Biomedical Engineering and a Mosaic K99 scholar. His current research is focused on how inflammatory cytokine signaling regulates fibroblast subpopulation dynamics in tissue fibrosis and using novel biomaterials to determine how that differs during tissue regeneration. Thank you very much for honoring our invitation. You can proceed with your presentation.
For that kind introduction. Like he said, my name is Daniel Abebayehu. And I wanted to take a second really quickly, just some news today. University of Virginia's dealing with the recent shooting, and I just want UVA or anyone you know and love is there reaching, and I'll make my talk today, but my talk myeloid-derived IL-1 signaling and how it can induce a unique population that appears to be critical. It recently been reported that approximately 4 developed world can be attributed to some sort of tissue.
Sorry, Dan. I'm so sorry. You have a problem with your microphone. We can hear it kind of, you have like a robot voice. If you could maybe switch or remove your AirPod. I don't know if it's possible.
Is this any better?
Sorry, repeat again.
Is this better?
Yeah, it's better, but there is a little lag, but I think it's better than previously. Yeah.
Okay. I'm sorry about that.
Very much.
Like I was saying earlier, approximately 45% of deaths within the developed world can be attributed to some sort of tissue remodeling disease. And in our lab, we're really interested in trying to understand some of the mechanistic insights that underlie various forms of fibrotic disorders. There's been a lot of work that has characterized and defined the inflammatory component of fibrotic diseases, as well as even more so the stromal compartment, trying to characterize what prompts aberrant fibroblast activation as well as dysfunctional mechanotransduction. But it's the interface between these two that's been rather understudied, and where I've tried to highlight the focus of my work. The disease that our group focuses on, or at least one of them, is idiopathic pulmonary fibrosis. It's a term disease that's been defined and characterized by excessive ECM deposition within the lung that inhibits alveolar movement, inhibiting eventual gas exchange. There's no good diagnostic markers. The current gold standard is radiographic imaging. And because of that, typically it's caught in the late stages of the disease, and that leads to median survival rates that are about 2 to 3 years, typically. In addition to that, based on the title idiopathic being the name of the disease, there's no known cause for the disease. There's no cure, and the currently FDA-approved therapies are only somewhat successful at slowing down the progression of the disease. So there's a huge clinical burden to try and better understand and address idiopathic pulmonary fibrosis, or IPF. What's really fascinating is that if you look at histological imaging of IPF lungs, you can actually see almost a time scale of the disease where you have normal lung to the left with intact alveolar structures. You see dense fibrosis to the far right. And in the middle, there's this unique, rather unique sites of active fibrosis And what's interesting is that typically from normal lung to fibrotic lung, it's an order of magnitude difference in stiffness going from about 2 kilopascals to 20. But these active sites of fibrosis are not very stiff. They're not too significantly different from normal healthy lung. But what's noteworthy is that these active sites of fibrosis that we have dubbed fibroblastic foci have fibroblasts that are not prompted by changes in stiffness, but rather some other mechanism by which these sites are the active sites of fibrosis. And previous work from our group has defined these sites as being both alpha SMA positive, the canonical myofibroblastic marker, but also CD90 or Thy1 negative. Thy1 is a rather pretty fascinating molecule. It was originally discovered on thymocytes or T cell precursors, but in the context of fibroblasts, they actually serve as a rigidity sensing molecule. It's a GPI anchored protein on the plasma membrane. And what we've seen is that it can confirm mechanosensitive cytoskeletal remodeling if you take mouse lung fibroblasts and seed them on either soft or stiff substrates to kind of match the biophysical environment of either healthy lung or fibrotic lung. Thy1 positive fibroblasts will undergo remodeling of their cytoskeleton to kind of respond and match their native environment or their environment that they're on for their substrate, with 2 kilopascal soft substrates having rather not noteworthy actin cytoskeletal stress fibers forming. But on the stiff substrates, you see these really noteworthy actin stress fibers appearing, as well as really large focal adhesions. But what's fascinating is that Thy1 negative fibroblasts, regardless of whether they're on soft or stiff, rather appear to have a cytoskeleton that has the same degree of stress fibers forming as well as focal adhesions, indicating that they don't respond to their mechanical environment as they typically do. And interestingly, when we knock down Thy1 using shRNA, we see via AFM, where you can basically just see the stiffness of a cell by tapping on it with the AFM. We see that on a stiff substrate, Thy1 positive cells will be able to respond to their environment. In the absence of Thy1, they're unable to. And finally, when we take these cells and seed them on different material substrates, we see that Thy1 negative fibroblasts are uniquely able to begin to produce and secrete new ECM based on fibronectin and procollagen 1 staining. And wanting to know what Thy1 does, we were able to do via IP show that it binds to inactive alpha V integrins, indicating that a key role for how these fibroblasts are responding and engaging with their environment is via alpha V integrin engagement. So the kind of the mechanistic model of all of this is that on soft materials or soft substrates, fibroblasts will have Thy1 engaging alpha V beta 3 integrins in a bent inactive conformation, and on stiffer substrates enable alpha V beta 3 to engage and bind to their local ECM, driving contractility and downstream signaling. Thy1 negative fibroblasts will have alpha V beta 3 constantly available to bind to their ECM and driving contractility and downstream signaling regardless of their the substrate stiffness. This is really fascinating because we're able to see that if you look around these fibroblasts, they seem to engage and strain stiffen their local ECM differently. And when we use an alpha V beta 3 neutralizing antibody, that goes away. And interestingly, in current models of lung fibrosis, usually we use a bleomycin to induce it. But what's interesting is that in the black 6 mouse, by day 42, the fibrosis goes away. In the Thy1 knockout animal, that fibrosis emerges and is progressive and does not get better. And these mice actually begin to die off. And we can see dramatic differences in alpha V beta 3 engagement, the actual mechanical resistance of the lungs over time, and that histologically over time, these Thy1 knockout mice get worse. One of the key questions that we've yet to answer is that while Thy1 absence is localized to these active sites of fibrosis, we don't know what promotes the loss of Thy1. And one of our emerging hypotheses is that based on the total lung having only a difference in 10% based on Thy1 expression in IPF, it's localized to the foci. And fascinatingly, the noted inflammation that we see in fibrosis, where you can actually use IL-1 beta instilled intratracheally to promote lung fibrosis. There's still this disconnect between the chronic inflammation in lung fibrosis and the stromal compartment that's characterized by adherent fibroblast activation and changes in mechanotransduction. And our thought process is that, or our hypothesis is, perhaps inflammatory cytokine signaling might be promoting the loss of Thy1, which then drives altered mechanotransduction that we see in IPF. So the way that we want to try and go about addressing this hypothesis is we looked at publicly available RNA-seq data sets to look at what cytokine receptors fibroblasts express, and then see if there are changes in receptor expression between normal or healthy lung versus IPF lung. And rather interestingly, what we're able to see is that in fibrotic versus non-fibrotic regions, that we saw IL-1 receptor 1 staining co-localizing with sites of really bright alpha SMA signaling, indicating that perhaps IL-1 receptor might be a signaling axis to focus on at these fibroproliferative regions within IPF. What we wanted to do next was use the bleomycin model of lung fibrosis to try and see if we take away, if we have a global knockout of IL-1 receptor, what happens? And in fact, interestingly, as expected, the Thy1 knockout mouse had a more severe, histologically severe form of lung fibrosis. But when in the absence of IL-1 signaling, we saw that there was really very little response to bleomycin, indicating that IL-1 receptor signaling loss is able to ameliorate bleomycin-induced fibrosis here. What we next wanted to try and figure out was where is the IL-1 ligand coming from, and this can be a little difficult because typically cells that are releasing IL-1 beta are cells that are undergoing cell death, and so it can be hard to identify them. And in addition to that, typically IL-1 beta is translated and stored in its precursor form, so it's hard to detect the transcript level. And what we were able to use was a reporter mouse for the inflammasome as a surrogate. What we were able to do is that there's an M-citrine fluorophore that was tagged to the ASC protein that's a component of the inflammasome. And so figure B over to the right here, you're able to see using imaging flow cytometry that when there's no inflammasome activation, the fluorescent reporter is kind of diffused throughout the cell. But when it's activated, there's this speck that forms that we're able to see which among these cells are SPEC positive that indicate the activated inflammasome. So we wanted to define what cells are SPEC positive that have the activated inflammasome. Using imaging flow cytometry, we were able to identify that the immune cell compartment was by far as we expected, by far the most SPEC positive inflammasome activated cell population. And when we broke that down further, we were able to see that it was the CD11B compartment, CD11B positive compartment that was had the most significant SPEC positive population. So for now, we can only conclude that this is myeloid positive, but we're hoping to do further analysis given that this is within the lung. We need to try and do more staining for cyclic F as well as CD64 to define whether it's alveolar macrophages, dendritic cells, or interstitial macrophages. Next, we want to try and see what is IL-1 beta doing beyond Thy1. And so we first wanted to look at specifically Thy1 loss, and we're able to see that treatment with IL-1 beta of these fibroblasts on soft substrates, these are 2 kilopascal hydrogels, we see a pretty noteworthy loss of Thy1 expression, and as expected, an increased activity in active alpha V beta 3. When you gate on Thy1 positive versus Thy1 negative, we're able to see that, as expected, Thy1 negative fibroblasts had more active alpha V beta 3, but rather interestingly, that the Thy1 negative fibroblasts also expressed far more IL-1 receptor 1, indicating that perhaps these cells, which are usually a very small proportion of the resident fibroblast population, are uniquely poised to respond to IL-1 receptor ligands and perhaps can serve as a bit of a unique inflammatory or immune responsive fibroblast subpopulation. what we next wanted to try and figure out was leveraging the fact that Thy1 knockout fibroblasts, Thy1 knockout mice have a non-resolving phenotype in the bleomycin lung fibrosis model. We wanted to see if we have a double knockout of Thy1 IL-1 receptor, what happens. So as I mentioned earlier, Thy1 knockout mice and wild type have at day 14 developed lung fibrosis in response to bleomycin. By day 42, wild type mice recover. Thy1 knockout mice continue to get worse. For the double knockout of IL-1 receptor 1 and Thy1, these mice display a less severe form of lung fibrosis in response to bleomycin, indicating that perhaps the loss of IL-1 signaling, even in a Thy1 deficient environment, is able to ameliorate a really traditionally severe fibrotic reaction. So in conclusion, we were able to see that IL-1 receptor 1 appears to be present within the fibroprolific region of the IPF, that these Thy1 knockout mice demonstrate very severe fibrosis, and that IL-1 receptor knockout mice display little fibrosis in response to bleomycin in this model, and that the IL-1 signaling is myeloid-derived and can promote the loss of Thy1. And finally, that the absence of IL-1 signaling is critical for the fibrotic phenotype that we see within Thy1 deficient fibroblasts. I want to thank the Barker Lab and everyone, all of our collaborators for this work. We're still trying to get this out there, but we're really excited about the story. I want to thank our funding sources, and I'm happy to answer your questions.
Thank you so much, Dr. Dan, for your presentation. We'll we'll receive questions now from our audience. Okay, so while we await questions from the audience, so I just wanted to ask you that you share maybe some words of advice for young and upcoming Black immunologists in the event who joins the event.
Yeah, the best piece of advice I have is find this kind of goes off nicely with I think some of the things that Dr. Nicholas, Dr. Butts was saying earlier, but I've found that having a community that you can kind of a local community that you can have to use as a source of either commiserating together or a source of getting encouragement is really huge. Over the years, I've kind of learned that science is far more relational than people typically describe science as. We always talk about scientists being, they're always stuck at the bench or we're lab rats, but we're mentored by people, we mentor people. There are people, we work alongside with, people we work on behalf of, people who suffer from the diseases we study. And so science is very relational, and I think it's important to find the relationships that are very life-giving, that empower you, that challenge you, that encourage you. And for me, I think a lot of any of the successes that I've had, I think are a byproduct of the really positive relationships that I've very positive relationships that I've had in the community that I've been able to establish.
Okay, thank you very much for that response. We have a question from Dr. Yassin. He's asking if interleukin-6 downregulates Thy1 as well.
That's a great question. So IL-6 it actually upregulates Thy1, interestingly, and so I think what we've tried to see is whether any of the transcription factors that IL-6 promotes activation of, if that same transcription factor binds to the promoter of Thy1. It seems like at the bioinformatic level it appears to be the case, but we hope to test that experimentally.
Thank you so much for your response.
Thank you for having me.
Thank you so much for joining this session, Dr. Daniel, and thank you to everyone who joined us, our audience. So we look forward to tomorrow's session, where we'll be discussing about immunology without borders, grounding yourself as a scientist and applying bioinformatics to your research. Do join us tomorrow.